Hi everyone, and welcome to Biology Professor. Today we're going to be talking about a specific laboratory test called the Snyder Test, which uses Snyder Test Agar to determine a person's cavity susceptibility. So first of all, let's think about what causes cavities. Well, a major cause of dental cavities, also known as dental caries, are oral bacteria, that's bacteria living in the mouth, that ferment dietary carbohydrates, so carbohydrates that you eat, particularly refined sugar, producing an acid that degrades tooth enamel. So basically, if you're eating a lot of candy, drinking soda, white bread, a lot of that kind of thing, a lot of carbohydrates in your diet, that provides a food source for these specific oral bacteria like Lactobacillus acidophilus and Streptococcus mutans. Those are two of the major ones, certainly not the only ones though, that will ferment those sugars, producing an acid, and the acid damages your enamel on your teeth. And so that brings us to the Snyder test. So a Snyder test uses Snyder agar to measure a person's cavity susceptibility. And it does this by taking an oral sample, basically saliva, and measuring the amount of acid produced by bacteria in the saliva over time. This Snyder agar in particular is known as a differential medium. Differential medium is one that visually distinguishes bacteria based on biochemical characteristics. So basically, if you've got different types of bacteria, they have different kinds of biochemical characteristics, such as the ability to ferment or not. That can result in changes that can be seen visually, something like a color change, for example. And in particular, with the Snyder agar, it has to do with the pH. And so the agar itself starts at a pH of about 4.7, and there is a pH indicator in there called bromocresol green, which is at this pH of, when it's at a pH of 4.7, it is green in color. But when acid is produced by these oral bacteria, things like this Lactobacillus and Streptococcus, that will lower the overall pH to 4.4, and anything that's 4.4 or lower will cause that bromocresol green to be yellow. So with that in mind, let's look at the test procedure. You start out with Snyder agar. It's green initially. The Snyder agar is composed of glucose. Why glucose? Well, we said that it's dietary carbohydrates that these bacteria ferment, and so glucose is the carbohydrate that's used in this test. And then I already mentioned the bromocresol green that changes colors based on the pH. It's our pH indicator. And then the Snyder agar also includes agar. That's just the solidifying agent. In other words, that's what keeps the stuff in the tube kind of this solid gel-like consistency instead of a liquid. And so here's the procedure. The person will have approximately 0.1 milliliter of their saliva collected. This is prior to dental care. That means prior to brushing their teeth. They wake up first thing in the morning and they collect a saliva sample before they've brushed their teeth or flossed or done any mouthwash to collect the bacteria that's collected in biofilms, that's kind of grown up as a biofilm on your teeth overnight. So get at least 0.1 milliliters of the saliva, inoculate melted Snyder agar. That's just because when you order the Snyder Agar, it's typically solid, so you loosen the cap, boil it for a while, that will melt it, then you can add your 0.1 milliliters of saliva to it, put the cap back on, kind of roll it in your hands to make sure that the saliva gets mixed throughout that melted liquid, and then as it cools to room temperature, the agar will re-solidify. You then incubate at 37 degrees, and that's body temperature, for 3 days. Some protocols call for 4 days, but I'll tell you why that doesn't really matter in a second. Then you need to interpret the results, and you do need to be checking the tube every 24 hours. So you can't just let it sit for 3 days. You have to keep checking on it at 24 hours, 48 hours, 72 hours, etc. And so you interpret the results based on this table. In this table, wherever you see a plus sign, that means that the tube has turned yellow. Wherever you see a minus sign, that means that the Snyder Agar has remained green. And so, of course, yellow means that the pH has fallen to 4.4 or below because of acid production during fermentation by these oral bacteria. And if it's still green, that means there's not enough of that bacteria in the oral saliva sample to create enough acid to cause the color change. And so you just look here. If at 24 hours, the tube is already yellow, meaning a positive sign, that means that the cavity susceptibility is considered severe. And I've seen this with students before. Their saliva turns yellow overnight, and they have a severe cavity susceptibility, and typically they've had a lot of cavities in their lives. If it's still green at 24 hours, but by 48 hours it's turned yellow, that's the moderate susceptibility. If it's still green at 48 hours, but turns yellow at 72 hours, that's the slight susceptibility. If it is still green at 72 hours, that's after 3 days of incubation, then we consider that a negative cavity susceptibility. And that's because by 96 hours, which is after 4 days of incubation, whether it's yellow or green, it's still really considered a negative cavity susceptibility because there were so few of these types of bacteria in the oral sample. So I want you to think about a couple of things here. If your sample turns yellow, immediately or within 24 hours, does that mean that you are doomed to having cavities? No, it doesn't, because remember that what causes the cavities is the acid that is produced only when these bacteria have a lot of dietary carbohydrates to be fermenting. So if you're following a low carbohydrate diet, you're eating few carbs, limiting your processed, refined sugars in particular, then even if you have the bacteria in your mouth, they're not producing this acid that will cause the dental cavities. On the other hand, just because you have a slight or negative susceptibility does not mean that your diet doesn't matter, because you likely still have at least a few of these. If you start eating a lot of these refined carbohydrates, they are likely to increase in amount in your mouth, which could lead to cavities later. Also remember that it's possible to not have very many of these in your mouth and then to suddenly get more of them in your mouth. For example, if you are eating and drinking after people or kissing someone, anything that would result in a lot of saliva exchange over time, you could possibly get bacteria from their mouth that then takes up residence in your mouth, and that then does increase your susceptibility to cavities. So if you are interested in this, you can also see some videos I have on other differential media. For example, mannitol salt agar is one that's another differential medium that I have a video on that you can check out if you are interested. Thanks for watching Biology Professor today, and have a great day.